Good afternoon, everyone. I'd like to start off by thanking you for your attendance today. My name is Jordan Young, and I'm the Strategic Marketing Manager here at IDT. It is my pleasure to be moderating today's XGen NGS webinar event in collaboration with the Munich Leukemia Laboratory. Before we begin today's presentation, I'd like to provide a brief overview of our guest speaker, Manja Megendorfer, Head of Molecular Genetics at MLL. Manja is responsible for research and development with an emphasis on NGS and leukemia diagnostics, especially with regards to the ongoing 5,000 whole genome projects aimed at a better understanding of leukemia's genetics. She studied biology at the universities of Tübingen and Munich with a focus on human genetics, immunology, and bioinformatics. She received her Master's of Science in Biology from the Ludwig Maximilians University in Munich and her PhD in collaboration with the Helmholtz Center Munich, working on gene regulation within the architecture of cell nucleus. After her postdoctorate, she moved to the field of hematology as a scientist in the Department of Molecular Genetics at MLL. Besides leukemia diagnostics, her scientific work focuses on mutational patterns and their prognostic impact within different leukemia entities. She has published and contributed to more than 50 scientific papers in hematology in the last 7 years. In combination with her MBA in healthcare management, the aim of her work is also to improve the diagnostic and prognostic work to optimize routine diagnostic approaches for patients with leukemia or lymphoma.
Also, a warm welcome from me. I look forward to sharing data from our lab with you on the topic The Future of Personalized Genomics, Advancing Hematology Research with NGS. For characterizing hematological malignancies, different analytical methods are used. Phenotypic characterization is based on morphology and immunophenotyping, addressing the cell counts, dysplasia, maturation, and antigen expression. Genetically relevant aberrations are investigated by cytogenetics, meaning chromosome bending analysis in fish, and molecular genetics. Here structural variants, SVs, copy number variations, CNVs, single nucleotide variants, SNVs, fusion transcripts, and differential gene expression are of interest. While until recently phenotypic methods were the decisive diagnostic factor, genetic methods and the genetic profile of the diseases are becoming increasingly important in order to enable targeted therapy. The analytical method behind genetic profiling is next-generation sequencing. Based on the identified variants, patients are classified into different risk categories and targeted therapy is enabled. This represents the concept of precision medicine. There are different approaches for sequencing. There is whole genome sequencing, which covers the cellular DNA almost completely and allows detection of nearly all kinds of variants. On RNA level, there is total RNA sequencing, which represents total cellular RNA and the respective gene expression, detection of fusion transcripts or splice variants. Then there is exome sequencing, which represents the coding region of the genome. Lastly, there is the option for targeted sequencing, panel sequencing, which covers the genes or fusions of interest, depending on the tumor type. Although the analysis of gene mutations in the standard procedure, there is today the option to spike in additional probes, like probes covering the complete genome and allowing CNV detection, copy number changes, and copy-neutral loss of heterozygosity, CNLOH calling. And that's why we use a targeted panel workflow for most of our research projects, aiming at the genetic characterization of different leukemias and lymphomas. We use currently different customized panels, as well as spike-in panels for specific research questions. With that targeted approach, we perform deep sequencing, allowing the detection of small cell clones in large cohorts. Our standard workflow includes an enzymatic fragmentation, barcoding, amplification, and a hybridization capture workflow for enrichment of the regions of interest. We have fully automated the library prep and enrichment, with the enrichment running overnight. Using different panels and spike-in panels allows combination of different samples and projects respectively within one enrichment run. We use currently different customized panels as well as a SNP ID panel and a CNV panel as a spike-in. Today I want to focus on 3 research projects using the targeted approach. The first one addressed in first line gene mutations by using the somehow biggest targeted panel, the exome panel. The second project targets on a genome-wide CNLOH detection using a targeted panel and a CNV spike-in panel. And finally, the last project addressed specifically mutations, deletions, and CNLOH of one target gene. The first project I want to present is the exome sequencing of MPN patients progressing to blast phase. 20% of patients diagnosed with a myeloproliferative neoplasm will progress to blast phase with dismal outcome. Our study cohort consisted of 19 patients, 8 female and 11 male, with a median age of 73 years. The median time between MPN diagnosis and MPN blast phase was 33 months. The blast count increased from less than 2% to more than 50% during progression. Cytogenetics was available in 15 of the 19 patients at both time points. In most cases, we had bone marrow material for our investigations. All patients showed an MPN driver gene mutation, 17 in JAK2 and 2 in MPL. In all 19 patients, we performed exome sequencing at both time points, MPN and blast phase, using our standard hybridization capture and enrichment workflow with the exome panel. The median coverage was 257x. We analyzed the genetic background at MPN diagnosis and blast phase separately, as well as in comparison. The calls for small nucleotide variants were filtered by a variant allele frequency above 5%, a GNOMED frequency below 0.005%, and an in-house developed score for a functional impact of the variant on the protein structure. Furthermore, we investigated the copy-neutral loss of heterozygosity by the SNP allele frequencies, as well as the copy number variations. Looking at the exome-wide mutation frequencies revealed that each patient harbors in median 81 and 80 variants at MPN diagnosis and MPN blast phase respectively. However, alterations occurred, since in average 9 mutations were lost and 12 mutations were gained during progression. Over all 19 patients, more than 1,000 different genes were affected at both time points, and only a small proportion of these genes, around 5%, were recurrently mutated, with more than 2 patients showing a mutation in a specific gene. These donut plots show the distribution of gene mutation frequencies. At both time points, more than 950 genes were mutated only once, while 60 genes in MPN and 81 genes in blast phase were affected by mutations in 2 cases, and only 52 and 57 genes were mutated in more than 2 patients respectively. Cumulating the variance of both time points per patient revealed that in median every patient showed 98 variants. 68 of these variants were unique and not detected in any of the other patients. Addressing the variant allele frequencies showed that in 40 of these unique variants, the allele frequencies remained stable. This indicates that half of the variants per patient are patient-specific and potentially not associated with the progress of the disease. When we looked at the functional annotation of the mutated genes, it became clear that genes belonging to the splicing machinery, as well as epigenetic regulators and chromatin modifiers, remained stable during progression without large differences in mutation frequencies between both time points. This is illustrated here by the comparable mutation frequencies shown by gray bars for MPN and by red bars for blast phase. In contrast, genes involved in the RAS pathway were more frequently mutated in blast phase than at MPN diagnosis. Genes highlighted in red showed an increase in mutation frequencies at blast phase, as illustrated by the height of the red bars here. Further analysis focused on the recurrently mutated genes. At MPN diagnosis, beside the MPN driver gene mutations in JAK2 and MPL, the most frequently mutated gene was SRSF2, followed by TET2, A6L1, RUNX1, and DNMT3A. As indicated by the light blue boxes at the bottom, most patients did not harbor any chromosomal aberrations, and only 2 patients showed an aberrant karyotype. The latter finding changed dramatically by addressing the karyotype in MPN blast phase. Here, 10 patients showed an aberrant karyotype, and for 50% of these patients, even more than 3 chromosomal abnormalities were detected, which is commonly referred to as a complex karyotype. Additionally, I want to draw your attention to the changed order of frequently mutated genes. At MPN blast phase, the most frequently mutated gene was SRSF2 due to the loss of some CHEK2 mutations. RUNX1 passed A6L1 and TP53 mutations appeared. In the next slides, I want to address the clonal evolution of these patients. Therefore, I would like to introduce the following figure. Here you can see the clonal changes of a single patient. The top of the graph reflects the clone at MPN diagnosis, while the lower part reflects the clones at MPN blast phase. The named genes and aberrations show the characteristic of the clone, and the width of the clones illustrate the clone's size. A red marked clone shows an increase in clone size during progression, while a blue one a decreasing, and the gray one a stable clone size. Gene names at the bottom of the graph illustrate gained gene mutations or aberrations. First, we addressed the CHEK2 mutation status at MPN and blast phase. In 6 of the 19 patients, the MPN-defining clone, characterized by the CHEK2 and MPL mutation, was completely replaced. In another 3 patients, the CHEK2 mutated clone was diminished. In the remaining 10 cases, the CHEK2 or MPL clone was stable or even increasing. This illustrates that not necessarily the MPN clone drives the transformation to blast phase. As I already mentioned, RAS gene mutation frequencies increased during leukemic transformation. As we investigated the recurrently gained gene mutations in more detail, we found out that beside the RAS gene mutations, more precisely 3 patients acquiring a NRAS or PTP11 mutation, seen here on the right side, also mutations in FLT3-ITD, CBP-alpha, and KM22A PDD were gained. These affected genes are typically found in de novo AML rather than in secondary AML patients. Even more often, mutations in RUNX1 were gained during progression to blast phase. While 3 patients carried a RUNX1 mutation from the beginning of the disease, 6 patients acquired a RUNX1 mutation during progress. And another 4 patients gained a mutation in TP53. No need to say that all these 4 patients acquired additionally a complex karyotype. Last but not least, we focused on the most frequently mutated genes in our cohort, SRSF2 and TET2. Noteworthy, 14 of the 19 patients showed a mutation in either SRSF2, shown on the left side of the slide, or TET2, seen in the middle of the slide, or in both genes, seen on the right side. Since these mutation frequencies were high, we compared these data to an MPN control cohort that showed no progression to blast phase in the follow-up. The mutation frequencies of SRSF2 and TET2 at MPN diagnosis in the control cohort was with 9% for SRSF2 and 17% for TET2, significantly lower compared to our MPN cohort of patients progressing to blast phase, showing 68% and 47% of mutated patients respectively. To summarize the results of the first project, by exome sequencing, we identified in median 80 variants per patient, whereof most of the variants were most likely not associated with the disease or disease progression. Blast phase can be driven by an independent clone. SRSF2 and TET2 are more frequently mutated in progressing MPN, and RAS pathway mutations are often gained during progression. Since the next 2 projects will focus on copy number variations and copy neutral loss of heterozygosity, I would like to briefly introduce the spike-in panel. Normally, CNV and CNLOH are addressed by chromosome banding analysis or array CGH. More recently, DNA-based assays like whole genome or exome sequencing allowed CNV calling. However, for targeted assays, CNV backbone panels that contain probes that are spaced approximately every 0.34 megabases across the human reference genome also allow the detection of CNV and CNLOH, enabling the screening of large cohorts with high sensitivity. Here you can see two examples of the obtained copy ratio plots. In the left plot, you see the gain of the complete chromosomes 12, 18, and 19. In the right plot, the loss of chromosome 7 and the short arm of chromosome 12 are visible. In addition to single gains and losses of chromosomes, polyploidies can also be detected based on the copy ratio. Here you can see trisomies for a number of chromosomes, also detectable in the variant allele frequency plot that normally differs from 50% at uneven copy numbers. However, some chromosomes are present with 4 copies, and here the variant allele frequency is 50%, since most often the diploid chromosome pairs are duplicated. So the differentiation between trisomy, tetrasomy, and pentasomy is possible based on copy ratio and variant allele frequency plot. However, I have to admit that using the copy ratio and the variant allele frequency has its limitations. For example, this is a complex karyotype that is easily identified as such, but not easily resolved by the copy ratio of the spike-in panel. But on the other hand, even the deletion of very small chromosomal regions can be detected, as seen here for chromosome 17p, where NF1 and SAS12 genes are located. The deletion was also visible by the reduced coverage of the targeted panel at that genomic region, addressing the gene mutation. Furthermore, here you can see the first CNLOH for chromosome 9p, where CHEK2 is located. A CHEK2 mutation in this sample appeared therefore with a high mutational load, illustrating the homozygous status of this CHEK2 mutation. But what is the biological mechanism for CNLOH? CNLOH results often in the duplication of an activating mutation in an oncogene, or the deletion of a tumor suppressor gene, or alters the specific site for any methylation. These 3 examples illustrate that in hematological malignancies, CNLOH is most often occurring in the combination with the gene mutation, resulting in homozygosity, like here for TET2 or EZH2 or DNMT3A and TET2. But coming to the second project investigating genome-wide CNLOH in a large cohort of more than 1,000 samples. We sequenced all these samples with a median of 1700x for the targeted gene panel and spiked in the CNV probes in a way that these regions were covered by median 50x. As a control cohort, we had the data of nearly 4,000 whole genomes. Most of the samples were analyzed with the myeloid panel, a smaller fraction with our lymphoid panel. We analyzed the single nucleotide variants and used the coverage information as input for CNV-KIT. Interestingly, 6% and 10% of the samples show at least one CNLOH, an aberration that so far was rarely investigated. This karyotype illustration highlights and summarizes the occurrence of CNLOH and the affected gene mutations. Interestingly, there were differences in the occurrence of CNLOH events and associated gene mutations between myeloid and lymphoid neoplasms. Some associations appeared in both myeloid and lymphoid neoplasms, shown here in purple, while others occurred only in myeloid neoplasms, illustrated in red, or lymphoid malignancies, illustrated in light blue. Finally, I will focus on my last project, the combination of variant and calls for addressing the mutation status of the tumor suppressor gene TP53. It's becoming increasingly evident that the mutation status of TP53 influences the prognosis of patients. The question is, are there prognostic differences between single or double mutated TP53? And which methods do I need to address this question comprehensively? The most popular methods in use are FISH and gene sequencing. We investigated more than 2,000 patients with a suspected MDS, myelodysplastic neoplasm, by targeted myeloid panel sequencing and investigated small nucleotide variants, copy number alterations, and CNLOH. We identified 185 cases with a mutation in TP53. A TP53 multi-hit status can be caused by a mutation and additional CNLOH, additional deletion, or more than one mutation. In our cohort, 24% were caused by additional CNLOH, 25% by additional copy number change, meaning deletion of 17p, and 51% by more than one mutation in TP53. Looking at the variant allele frequencies of the affected mutations showed a higher variant allele frequency with additional 17p deletion or CNLOH. However, the cases with CNLOH could not be identified by a high VAF only, since the VAF overlapped also with other constellations, illustrating the importance of CNLOH detection beside detection of mutation and 17p deletion. And with that, I would like to summarize that NGS plays a pivotal role in genetic profiling and therefore personalized medicine. Targeted panel sequencing allows a deep dive in genetic research questions. Spike-in panels like the CNV backbone panel broaden the readout significantly. Regarding our research projects, I would like to conclude that exome sequencing illustrated the diverse clonal evolution during disease progression. Genome-wide CNLOH assessment showed a number of recurrent events in hematological patients. CNLOH information cannot be replaced easily by variant allele frequency of the affected gene mutation and therefore contributes to the mutation status. Thank you for your attention. I'm happy to take questions.
On behalf of the IDT team, I'd like to extend a big thank you to our guests who attended today's event, as well as to our guest speaker, Manya, and the broader MLL team for their continued partnership and collaboration with IDT. Now, for guests that may have questions with regards to today's presentation or products, I encourage you to submit your questions via the Q&A portal so that one of the IDT representatives can assist you with those. Thank you, and I look forward to seeing you at our next event.